Here we are in the third tide pool down in the tidal height transect at Giant Stairs. And as we get closer and closer to the open ocean, the biological diversity is increasing. And so if we look at this tide pool, we can see many more species of algae. We have green algae, we have red algae, and we have brown algae living in this tide pool. We have many more mobile consumers here. We have limpets, we have slipper shells, we have a lot of Littorina littorea, we have some crabs. Often in this tide pool we see lots of anemones. So this tide pool has a lot of biological diversity, lots of consumers of oxygen and lots of generators of oxygen. And the way we test and measure the quadrats in this tide pool is we take our quadrat and we haphazardly toss it into the tide pool, and then wherever it lands we just push it to the bottom of the tide pool, and that is where we measure. So in this quadrat you can see we have some barnacles, Semibalanus balanoides, we have some green algae here, and then we have some sun-bleached Chondrus crispus here, and then this red is Petrocoelus, which is a red algae covering. So we will record the biological data. Oh, and here we have a Littorina littorea crawling around, and another one here. So we will record the mobile consumers, and then we'll record percent cover of the rest of these sessile or anchored species in the biological data sheets, and we'll compare over the tidal height in the different tide pools. Yeah, great.